Hello everyone and welcome back. In this section, we will briefly look at how proteins are made. I will avoid the details of this process and I will focus mainly on the principles that are involved. So let us begin. As you know, our genes are stored in the form of DNA in our cell's nucleus. This DNA will act as a template in order to make copies of itself. You should know that this process is controlled by enzymes, the names of which I will ignore in this lecture. mRNA is made in the nucleus. This is the messenger RNA. The M here stands for messenger. They carry messages that are encoded in the DNA. The protein synthesis actually occurs in the cytoplasm, and this process occurs with the help of ribosomes. The bases involved in RNA are slightly different to that of DNA. If you recall, there are 4 bases that DNA structure is made of, and we mentioned that they are A, T, C, and G. The only difference that we have in RNA is that the base U, which represents uracil, is used instead of T, which represents thymine. What happens when the mRNA is made is that the mRNA, which is the messenger RNA, makes a complementary copy of the DNA template. So if the code for DNA is, for example, ATG, then the mRNA will make a complementary code to that, which in this instance will be UAC, because the complementary code for A would be T. However, as we mentioned, in RNA, instead of T, we have U, which represents uracil, and the complementary code for T would be A, while the complementary code for G would be C. So that was a basic overview of the process. Now let us go into a bit of details by looking at what occurs in 2 of the main steps that are involved in protein synthesis. These steps are called transcription and translation. Transcription. In this process, what happens mainly is that a messenger RNA is made from a template DNA strand. So that is basically what this step is all about. Firstly, the double-stranded DNA splits into 2 single strands. Each of these single strands will act as a template. An enzyme will facilitate this process. It will cause the DNA's double helix strand to split. The messenger RNA is then made by copying one of the strands. When the copying is completed, the messenger RNA is then carried to the cytoplasm. We can probably compare the role of the messenger RNA to that of a person who delivers mail, because the messenger RNA is really carrying a message that it collected from the DNA. The following diagrams will summarize the process of transcription. First, we have a double-stranded structure of DNA. The enzyme will split the double strands apart. And finally, a messenger RNA, or a copy of the DNA, is made. The next process that is involved in protein synthesis is called translation. Translation occurs on the ribosomes, that is found freely in the cytoplasm, or they can be attached to the rough endoplasmic reticulum. Here is where protein synthesis really occurs. As the name suggests, translation is where the message from the mRNA is read and interpreted. When the messenger RNA is released from the nucleus, it will attach itself to a ribosome. tRNA, which is called transfer RNA, will then come in, and it will bring an amino acid with it. Look at the diagram on the right. The tRNA is the flask-shaped structure. At the top, the round structure that you can see there, these are the amino acids. Each amino acid will have a specific code, a triple codon. The triple codon at the bottom will come in sequence and match with the code of the mRNA. That straight line at the bottom that you can see there, that would be the messenger RNA. As you can see, the codon AAA would match with its complementary pair UUU, which is found on the mRNA. The second triple codon will then come in a similar manner, and then that will cause a chain of amino acid to form. It is called a polypeptide chain. Later, when the entire message is fully translated, the polypeptide chain will fold and form into a specific protein. Here's another diagram that is showing us how translation is taking place. We can see at the bottom we have the mRNA, which contains triple codons. When the tRNA comes in, then a complementary pair is made, and that causes the amino acid chain to form. So to summarize, two processes are involved in protein synthesis. In the first process, which we call transcription, a messenger RNA is made using a DNA template in the nucleus. In the second process, which is called translation, a tRNA would bring in an amino acid which contains a triple codon. This allows the formation of polypeptide chains and eventually a protein. If you have any questions regarding protein synthesis, please do not hesitate to ask. Thank you for listening once again.